Stability testing protocols. Here, stability testing of new drug products and substances comes under ICH Q1A guidelines. Means this stability testing of new drug products comes under ICH Q1A guidelines. Stability testing. What is stability testing? Here, stability testing is mainly used to determine whether the quality of a drug substance or product is changed over time by various environmental factors such as light, temperature and humidity, etc. Stability testing is nothing but we are performing stability testing on any drug product or drug substances to know whether the drug quality or drug safety or efficacy may changes when there is change in environmental factors such as light, temperature and humidity, etc. Stability testing protocols mainly used to provide evidence how quality of a drug substance or product change with various environmental factors. Here mainly these are the stability testing protocols such as general testing, stress testing, selection of batches, container closure system, specifications, testing frequency, storage conditions, stability commitment, evaluation and statement and labeling. These are the different types of stability testing protocols. These are the different stability testing protocols which mainly provides an evidence. This is the evidence. Provides evidence how the quality of drug substance or drug products change with various environmental factors. For example, if there is a change in environmental factors such as temperature or humidity or light, if there is increase in temperature, there is a chance of occurrence of degradation. If any product occurs degradation, it may be due to change in these reasons. Due to these factors, there is a chance of enhancing degradation. General testing. Here, information on stability of drug substance plays an important role in evaluation of stability studies. And next one is the stress testing. Here, stress testing of drug substance mainly helps to identify degradation products. Here mostly when the degradation products are present in the drug substance or drug products, by performing stress testing we can identify the degradation products in the drug products or drug substances. And stress testing mainly used to validate stability indicating power of analytical procedures used. It mainly done by using analytical procedures. Stress testing is mainly carried out on single batch of drug substance. Here we are performing stress testing on only one batch, single batch. And testing of stress testing includes effect of temperature. Here these stress testing mainly done by including effect of temperature such as by increasing 10 degrees Celsius increments such as 50 degrees Celsius, 60 degrees Celsius. If the temperature is increased above 60 degrees Celsius, it is for accelerated testing. And when the humidity is, for example, 75 degrees relative humidity, if the humidity is greater than 75%, it may leads to oxidation photolysis in drug substances or drug products. And next one is the selection of batches. Here in selection of batches in the stability testing, first we have to take at least 3 primary batches for stability testing of drug product or drug substances. From these 3 primary batches, we select one batch for marketing or one batch for manufacturing of production, large scale production. Because from these among 3 primary batches, we should perform stability studies on 3 primary batches. The batches which produce the result, it should be same formulation and in same container closure system as proposed for marketing. The manufacturing process of primary batch which is used for production batch should provide same quality meeting specification intended for marketing. Here the manufacturing process which is used for the primary batch, same procedure should be used for the production batch, and the products which are produced from the production batch, it should provide same quality and it should meet specification intended for marketing. It should meet the specifications and it should have same quality and same efficacy of that of primary batches. Next, the manufacturing process which is used for production batches should be provided same quality meeting specifications intended for marketing. Here 2 batches among 3 primary batches. First two batches are the pilot scale batches and the second one batch is the smaller batches. If these batches are justified, in that case the primary batch, the date of primary batch represents quality of entire product. Next one is the container closure system. Here stability studies conducted on dosage form packed in container closure system is same as proposed for marketing. Specifications. Here specifications is the Q6A guidelines comes under specifications. Here stability studies including testing of those attributes of drug products. Those are sensitive to change during storage conditions and which may lead to change in quality, safety and efficacy. Here in these specifications it mainly used to testing of those products which change their physical properties such as physical properties or chemical properties and also their quality upon changing storage conditions, such as testing of drug products. If there is a change in storage condition of drug product, it may lead to change in quality, safety and efficacy of the entire product. So when there is small change, when the small change in storage conditions of the drug product, it may lead to change in quality, safety and efficacy of the entire product. So testing should cover, this stability testing should cover all the properties such as physical properties, chemical properties, biological properties, microbiological properties, preservative content and functionality test. If the stability studies passed out all these properties, all these tests, in that case only we can introduce our product into marketing. In that case only the product can get approval from the regulatory bodies for marketing. The acceptance criteria for release and shelf life specifications are mentioned in ICH Q6A guidelines and ICH Q6B and ICH Q3B for specification of degradation product in drug products. Shelf life acceptance criteria derived from stability information, and here these shelf life acceptance criteria is mainly derived based on, it mainly explained based on the stability information of all products. Testing frequency and storage conditions. Here testing frequency is nothing but it should be done it should be done in alternate manner, such as for every 3 months or such as for every 6 months or yearly. For example, for long term stability studies, accelerated stability studies and intermediate stability studies. For long term stability studies, the stability studies should be carried out for every 3 months for first year and for every 6 months for second year and for every year for third year throughout the shelf life until the expiry date occurs. The stability studies should be carried out for every year for after third year of manufacturing. For accelerated stability studies, the stability study should be performed within 6 months of study. Here minimum 3 points, we should include 3 points, that is 1, 3, 6. These are the time intervals. At these time intervals, initial and final time intervals, at these time intervals we should perform the stability studies. These studies are known as accelerated stability studies. This is the 0, initial, 3 and 6 every 6 months. Intermediate stability studies, here 4 points included to T initial and T final, that is 0, 6, 9 and 12. This is the initial time and this is the final time interval. And next one is the storage conditions. In storage conditions for long term, accelerated and intermediate conditions are mentioned here. That is for long term 25 degrees Celsius plus or minus 2 degrees Celsius and 30 degrees Celsius plus or minus 2 degrees Celsius, and the relative humidity of long term is 60% relative humidity plus or minus 5%, 65% relative humidity plus or minus 5%. And for accelerated stability testing of storage conditions is 40 degrees plus or minus 2 degrees Celsius, 75% relative humidity plus or minus 5%. And for intermediate 30 degrees Celsius plus or minus 2 degrees Celsius, 65% plus or minus 5% relative humidity. If any significant changes, if any changes occurs in the accelerated studies, we can go for intermediate testing. And here the storage conditions of refrigerators is for long term and intermediate and accelerated 5 degrees Celsius plus or minus 3 degrees Celsius for long term refrigerator conditions, and for intermediate there is no intermediate refrigerator conditions, and for accelerated refrigerator conditions 25 degrees Celsius plus or minus 2 degrees Celsius, 60% relative humidity. and for freezer 20 degrees Celsius plus or minus 5 degrees Celsius, and there is no intermediate and accelerated storage conditions for freezer. And storage below minus 20 degrees Celsius, these can be case by case basis. Based on case by case basis, we can determine the storage below minus 20 degrees Celsius. And stability commitment. Stability commitment is nothing but when long term stability data on primary batches does not cover the shelf life proposed. If it does not cover, if long term stability data does not cover the shelf life, in that case they should make a commitment. They should make a commitment to the commitment to continue their stability studies and post approval in order to finally establish shelf life. For establishing shelf life, they should make a commitment for continuing their stability studies and post approval. If submission include data of production batches, if at least 3 batches should be submitted. Commitment should be made to continue long-term studies till proposed shelf life and accelerated studies for 6 months. If the submission of production batches is less than 3 production batches, in that case commitment should be made to study remaining batches. Here only 2 batches are submitted. Here in that case we should made a commitment to complete the remaining batches within long term and proposed shelf. Within the proposed shelf life and accelerated studies, within 6 months we can complete the remaining production batches, and we should submit the commitment. Such commitment should be made to the higher authorities. And if no production batches, if among 3, if no production batches is submitted, in that case we should make commitment to place first 3 production batches on long term through proposed shelf life and accelerated studies. Stability protocols used for studies on commitment batches should be same as that for primary batches. Here the commitment batches and primary batches, the stability protocol should be made same for commitment and primary batches. If it is not same for commitment and primary batches, in that case we should make justification, because we should give the explanation for that reason of the. Next, evaluation. Here checking of entire data is known as evaluation. Evaluation is nothing but confidence limit. It should be greater than 95%. If it is present greater than 95%, in that case only the batch is accepted and approved by regulatory bodies. If it is less than 95%, it should be read for retest. Retest is nothing but it should be sent for retesting. Again the all stability testing should be performed on that batch. If it is less than 95% of confidence limit. If it is greater than 95% of confidence limit, in that case batch is accepted and it should be approved by regulatory bodies for marketing. And stability labeling. After completion of evaluation, we should make the stability labeling, such as by national regulations, such as it should be stored in a dark place, it should be stored in room temperature, etc. Or the statement labeling should be done by national regulations. Finally, instructions are written in the form of statements on the drug product or drug substances.